A quick introduction about myself. So you might be wondering why a investigator at the Perlmutter Cancer Center is talking about SARS-CoV-2. And I think, like many of us at the start of this pandemic, we're kind of searching for ways to apply our particular skill sets and interests to the problem of the COVID-19 pandemic. And one of my interests as a cancer researcher has been in the area of functional genomics. And so my talk will be focused on how, through a number of great collaborations, we were able to leverage some of that expertise in our lab to try to learn more about these viruses and how they interact with cells. So the motivation for this research was to see if we could understand ask the question, can drugs that target host cell proteins potentially inhibit SARS-CoV-2 infection? Identify what are the host proteins and pathways that SARS-CoV-2 requires to successfully infect a cell. And to ask the question, could certain antiviral host factors explain the differences in outcomes among infected individuals? And so as someone who really focuses on therapeutic development, I thought about this problem as a traditional target discovery and prioritization pipeline, in which you start with identifying the potential target landscape and then move on to hit validation, lead optimization, and potentially early phase clinical trials. And so the way that we thought about this through the lens of viruses is obligate intracellular parasites that require host factors to complete their life cycle. And I think this audience appreciates, and anyone can appreciate, that viruses and SARS-CoV-2 in particular need to interact with cells. And maybe the simplest way to think about that is the interaction between spike protein and the receptor ACE2. And this transaction is important for the internalization of the virus and the initialization of infection. But of course, this process is actually much more complicated, and viruses interact with their hosts on many different levels. Some of these interactions are mediated by protein products that are encoded in the virus's genome and expressed inside the cell. And many of these are through co-option of normal cellular processes, such as translation, assembly, and trafficking throughout the cell. You can imagine that you'd like to understand exactly what each one of these transactions is between a given pathogen and its host, because each one of these steps in the life cycle may represent a liability for these viruses that you could potentially interfere with using a new therapeutic strategy. And so I think most scientific audiences, and this audience in particular, know this technology well, but we thought about ways to interrogate all of the ways that viruses interact with their hosts using a gene disruption approach. And so we focused on one technology, CRISPR-Cas9, which allows us to disrupt every gene in the genome by programming a programmable nuclease using a library of RNAs that target it to specific loci, and then can lead potentially to frameshift mutations from non-homologous end joining. And so the general anatomy of the genetic screens that we undertook look like this. We start with a susceptible cell that expresses just the Cas9 nuclease and infect that cell line with a library of viruses, each encoding a different sgRNA at an MOI such that each cell gets one sgRNA and then loses one gene. So we're effectively mutagenizing a large population of cells. And then we apply selective pressure by infecting with SARS-CoV-2 or other pathogens. And this can cause a shift in the representation of any particular sgRNA. In this instance, I'm showing an increase in representation of these red and blue sgRNAs, which in this instance would target genes that are essential for the virus to infect cells. And so this isn't a revolutionary idea. This is a well-trodden path for other pathogens, and many other labs realize that this would be an important way to use sort of shotgun genetics to identify host factors for SARS-CoV-2. And recently, a rich literature in the preprint and peer-reviewed literature started to emerge identifying what these host factors are. One of the gratifying things about seeing all these papers come out is how remarkably consistent they are with each other from lab to lab, even operating in a vacuum. And so that gives us a lot of confidence in some of the results that we're seeing. So our lab took a multi-scale screening approach. One approach is basically to use libraries that intend to target every gene in the whole genome and to try to survey all potential host factors. A second approach we took was to generate new libraries that react to a paper published by Neve and Krogan's group that identified through proteomics a protein-protein interactome between host proteins and virus proteins, and screen only this small subset of 332 genes to identify which of these putative interactors were functionally important for infection. And then the third body of work that's unpublished is we have custom libraries that focus only on the portion of the human genome that is thought to be druggable. And we've been screening this at high coverage under a number of different conditions to potentially identify candidate drug targets. So I just told you about the libraries. These are an important component of doing these screens, but the other important component is the cell lines that you choose to do the screens in. And so this is a very busy slide, but the point of this slide is to illustrate that we explored many different variables. So we looked at 7 different cell lines, each one modified by addition of ectopic ACE2 expression or ACE2 plus temporis. We infected them with virus produced on 3 different cell lines and at 2 different temperatures. And we put a lot of work in up front to identify a cell line that had ideal infection conditions for our screen. And we ended up settling on HUH 7.5 for a couple of reasons. One is that it can be infected without adding additional ACE2. And another reason is because it can be infectable not just by SARS-CoV-2, but by 3 other coronaviruses, NL63, OC43, and 229E. The other important thing about credentialing this cell line for our screens is to make sure that the reason that these cells are no longer stuck to the plate here isn't because the cells are secreting some lethal factor in response to infection, but rather that they're all getting infected with cells. And we immunostain for SARS-CoV-2 N protein to show that there's a high level of lytic infection in these cells. And this makes them suitable for the types of screens that we were interested in doing. But why were we so focused on screening against 4 different coronaviruses? And part of this is just, due to feeling like wanting to be completists. But the other reason for this is thinking about how these coronaviruses emerge in nature. And so we screened these 4 different coronaviruses, 2 alpha and 2 beta, with the idea that finding common host factors between distantly related coronaviruses might help us find host factors that would also be relevant to even further distantly related coronaviruses that may emerge in the future, since we know that there have been 3 deadly coronavirus spillover events in this century alone. And so this has been a highly collaborative project between 3 institutions in New York, at Memorial Sloan Kettering, Francisco Sanchez Rivera and Yadira Soto Feliciano made reagents for this project. Screens were done in Charlie Rice's lab at Rockefeller by Bill Schneider and Heinrich Hoffman. And then these materials then got shipped downtown to NYU, where we made libraries and did a lot of the analysis. So starting off with the work that we did focusing on the whole genome. One of the things that we're always very concerned with at the beginning of doing a functional screen like this is trying to understand whether the screen is actually working in the cell line system that we're interested in, and whether we see any signal that's related to the selective pressure that we're applying. So for each gene in the library, we calculate a fitness score, and that's what's shown on this scatter plot. And on the x-axis, we show a fitness score that results from knocking that gene out and then just carrying that culture forward. But on the y-axis, we show the fitness score in the presence of an active SARS-CoV-2 infection. So the internal controls for these screens are, we know which genes are generally not essential for life and which genes in the library are generally essential and so have a deleterious fitness effect, which are shown in red. So we see a nice separation between the blue negative controls and the red positive controls. So this tells us that overall the genetic screen is working. But the things that we're interested in are these purple dots, which are genes when they're disrupted, give a positive selective advantage to cells or a fitness advantage, and likely constitute potential host factors. And also on the opposite side of the spectrum, these yellow-orange dots could be potential antiviral host factors. The other thing that we always get concerned about in these screens is making sure that we're identifying biology that we expect to see. And so looking at these 4 different viruses, which are shown in the rows, and a few different known and putative host factors for these different viruses, we checked to make sure that we were finding what we might expect. So we do find that ACE2 is a hit for SARS-CoV-2 and an L63 and as a known proteinaceous receptor. 229E also has a known proteinaceous receptor and PEP, which we clearly identify as top hit. And of course, cathepsin L, which has been identified by multiple other groups. So this tells us that the selection we're seeing is also probably selecting for the phenotype we're interested in, which is resistance through loss of an essential host factor. So this is what all the data looks like for SARS-CoV-2. Again, each dot here is a gene in the genome that we're targeting. So I'll skip that slide because it seems to be killing the presentation software. But I'll say that we found about 200 putative host factors and then focused on hit prioritization using secondary screening by one-to-one validation. So in these series of experiments, we start with a susceptible cell line, once again, plate them out into 96-well plates, and then either transfect in an siRNA or a CRISPR RNA, and then infect with one of our coronaviruses and stain for either dsRNA or a coronavirus protein. And these are really helpful for identifying, kind of validating some of your top hits. We were able to identify a number of interesting ones that I'll talk about in a second. But one of the things that we learned along the way is that many hits that were identified in our screen and subsequently validated in clonal knockouts by other groups were not strong hits in these screens. So while these are essential to do and useful, if a hit from a pooled screen doesn't validate an arrayed screen, that can be due to the type of knockdown that you're doing or the limitations of the time that the screen allows you to do. So the other thing I'll point out here is we did validate in this approach a number of interesting hits. One of them that was required for all 4 of these coronaviruses, TMEM41B, was one of the top hits and a protein that we knew very well because we had been studying it for the last 5 years as a pan-flavivirus host factor. And this protein, NVMP1, have been implicated as having roles in lipid mobilization at the early stages of autophagy. And so we were already kind of thinking about the importance of this protein. And this ended up being the kind of the centerpiece of our primary paper published on this subject. But I'm not going to say anything more about it because Heinrich Hoffman and Bill Schneider, 2 of the first authors of some of these papers, will be presenting on April 14th in this forum on CRISPR screens identifying TMEM41B. And they're going to take a deep dive into the mechanistic biology that underlies TMEM41B as a host factor, both for coronaviruses and for flaviviruses, but not in more than 10 other viruses that we've tested. So quite a coincidence. So this was the slide that crashed before, so we're going to try again. Each one of these dots here is showing a different host factor that we identified in the screen. And one of the thing that I want to point out in this slide is that there's some remarkable internal consistency within these screens in which we can identify, in some cases, whole pathways that are elaborated by their positive selection after loss of that gene. So I'll just highlight 2 pathways, but there are more in the paper. One is heparin sulfate biosynthesis, and the other is control of lipid biosynthesis through SREBP signaling. And so these two pathways were both highly enriched, and we found that essentially every member of this pathway that doesn't have a buffering homolog was identified in the screen. And so I'll go through a couple of these quickly. So starting with SREBP signaling, so this is a membrane-bound transcription factor that is highly regulated by the presence of cholesterol using SCAP, this sterol sensor, which controls the localization of SREBP, and thus its exposure to two different sequential proteases that are required for its activation and trafficking to the nucleus. And what we found in our screen was that all four of these genes that are required in regulation of SREBP activation were very clear hits in multiple different coronaviruses. And one of the interesting things about this is that one of these proteases can be inhibited with a small molecule. And this sterol sensor has a tool compound for inhibiting its activity. The other pathway I told you about that was highly enriched was heparin sulfate biosynthesis. And so we found, so heparin sulfate are these very complicated biomolecules that get synthesized by a long enzymatic process involving multiple different enzymatic steps. And we found that several of these steps were inhibited along the way, and that one of these can be inhibited by suramin, which is a WHO essential medicine that has been given for over 100 years. One of the interesting things about this to us that I'll point out a little bit in the next slide is that it's known that heparin sulfate is required for SARS-CoV-2 to bind cooperatively to ACE2 on cells. And it's intriguing to think about how the specific sulfation patterns on these heparin sulfate proteoglycans might mediate that binding. So what I showed you so far for SARS-CoV-2 was really just focused on one virus, but excuse me, we also are able to organize these host factors into functional neighborhoods that tell us about the functional relationship between the different hits in terms of their biology. And these are useful for relating, the function of genes that you may not know about, but they're also useful for understanding the common and different relationships between different viruses and their host factors. So for example, we can see here that heparin sulfate biosynthesis is clearly required for SARS-CoV-2 in purple and for OC43 in blue, but not the other viruses. And this is quite interesting because SARS-CoV-2 and NL63 share a proteinaceous receptor, which is ACE2. So it appears that NL63 can bind to ACE2 independently of heparin sulfate. The other thing that can be quite interesting about these kind of diagrams is that it helps you to identify host factors that are required for many different viruses, just by the contribution of the different colors of the viruses to these pie charts. And so we were able to identify a number of these, including BMP signaling. I told you about TMEM41B, but these comparisons are useful when you really dig into these kind of pathways as a whole to include some of the genes that were not hits. So this is a heat map that's showing the requirement for different members of the heparin sulfate biosynthesis pathway. And as I mentioned, for these two coronaviruses on the far right, there's essentially no red, which means that there are no steps in this biosynthetic pathway that are important for either of these viruses. Whereas for these other viruses on the left, HCOV, OC43, and SARS-CoV-2, there are a number that are in common, but interestingly, HS2, ST1, which was the druggable one I told you about, is curiously not required for OC43, but is for SARS-CoV-2. And this is intriguing to me because it makes me think about whether the specific sulfation pattern imparted by this enzyme might be particularly important for SARS-CoV-2. Those were the findings from our whole genome screen. We also looked at, as I mentioned, targeted screens of putative protein-protein interactome. And we learned a number of things from this. One thing we learned is that these protein-protein interactomes really are enriched in functional host proteins. We also learned that some of these proteins, such as NSP7, appear to act as hubs for the more functional proteins and are enriched in specific hits. ORF9C and NSP8 are other examples of this. So this, I think, speaks to the value of doing these protein-protein interaction studies, and we were able to functionally validate a significant number of these putative interactions from the Krogan lab. The other thing that was quite interesting is when we looked at the cumulative distribution of z-scores for different viruses such as 229E, OC43, and NL63, they were also enriched in functional host factors that were identified from the SARS-CoV-2 protein-protein interactome. So this implies that there are potentially conserved protein-protein interactions that have a functional importance between SARS and other coronaviruses. And when we went back and looked at our genome scale data, one of the things that we learned is that while we found that some of the putative interactors were enriched in the genome scale screen, the interactors that we identified as being functionally important in this paper were even further enriched. So lastly, with my remaining time, I'll talk about some very early data looking at some of the druggable genome. So this is focusing on just a tiny sliver of the genome, so we can do a lot more screens and under a lot more conditions with much more throughput. So these are some of the early returns from this, and we're still trying to kind of work up the mechanism for some of these potential hits. But we found in red are potential host factors that are required for infection for these for these 3 different viruses, SARS at 2 different temperatures. And happily, we find ACE2 is found in these screens, which we would expect because it happens to be in the library and druggable. We find some other things in here that are nice as well, including a couple of different kinases, such as casein kinase and some SRC C-terminal kinases, and some kinases in the BMP signaling pathway. Also, one of the top hits for a couple of these was EF4A, which is a eukaryotic initiation factor that's important for resolving G quadruplex structures in RNAs. And this is was really nice to see because other groups, including the Krogan lab, identified through orthogonal approaches that this is probably a bona fide druggable target for SARS-CoV-2. And so what we're hoping is that in our druggable genome data, there may be some other targets that validate as well as EAF4A1 has. So to summarize, during this study, we performed hundreds of host factor screens for SARS-CoV-2 and 3 common cold coronaviruses. Our results are among the most comprehensive picture of coronavirus host transactions to date. We discovered multiple enriched pathways with translational opportunities for small molecule inhibition. And it does appear that PPI networks contain many putative host factors that are not required functionally. However, they're clearly enriched in those that are. We learned some lessons along the way. One is it pays to do the work up front to choose the cell lines for your screen with great care. It can be very informative to screen multiple related viruses, and this can illuminate important host factors that are shared between the viruses you may not even have emerged yet. We learned that these secondary one-to-one validation screens are essential to do, but they're fraught with their own experimental biases and limitations. We found that these focus screens can be useful, particularly when you have screens that are dominated by a single biosynthetic pathway. And I didn't touch on this much, but we also found that seeing isn't necessarily believing, and that sometimes when we thought a screen was going to go very badly based on what we were seeing in culture, it actually worked very well, and vice versa. Sometimes when we thought we were getting appropriate levels of selection, the screen went poorly. So whereas we started thinking about target discovery, we're now thinking more about hit validation, making clonal knockouts, and really digging into the mechanism of these different targets that we've identified. And before I skip to the acknowledgements, I just want to shamelessly plug that my lab is recruiting postdocs and technicians. And if functional genomics is something that interests you in cancer or in viruses, please feel free to contact me. And I just want to say, I mentioned this sort of at the beginning, but this has been a great collaboration between Charlie Rice's group and mine over the last 5 years, as well as Scott Lowe's group and David Alice's group at Rockefeller and Memorial Sloan Kettering. And I hope I've acknowledged a lot of the important contributors along the way. So with that, I'll stop and take any questions that people may have.
Excellent. Thanks, JT. I think you win the prize for the smoothest restart after a PowerPoint failure. Kudos to you. If I could lead off with a couple of questions, and again, I'm going to ask folks to raise their hands so I can call on you. You had mentioned the heparin biosynthesis pathway and the SREBP signaling pathway as potential targets. And you noted that there were a couple of small molecule inhibitors of those pathways. And I'm just wondering if those have been tested against COVID-19.
That's a great question. So some of them have been tested. I know that the protease has some activity, and I know that the protease inhibitor has some activity, and so does a few other targets. I think one of the problems is that a lot of the inhibitors that have been tested are fairly dirty tool compounds, and so if you use them at a high enough concentration, you can sort of have a lot of off-target effects that also inhibit virus infection. So in one of the papers I highlighted at the beginning from Neville Sanjana's group, they did a nice experiment, which was to go and knock out the putative target of a small molecule inhibitor, and then see whether giving the drug still killed the cells, and it did. So that suggested that there was probably an off-target liability that wasn't really known about those. So I think that none of these are what I would call none of the ones that I mentioned at the beginning there were sort of what I would call high impact or kind of clinical candidates that are well understood. I think that the heparin sulfate biosynthesis one, Suramin, is one that we still need to dig into more detail. That one has not actually been tested yet as a small molecule inhibitor, but we have a small project to get that going now that we have sort of the functional genomics stuff tied up.
Okay. And the other question was with respect to the heparin sulfate. So if I understood you correctly, ACE2 is being modified by heparin sulfate, and that's affecting SARS-CoV-2 versus NL63 differentially. Can you comment a little bit more on that? And I'm wondering particularly whether binding studies have been done on heparin sulfonated versus non-sulfonated receptor.
Sure. So I'm not an expert in the structural biology of that interaction, but my understanding is that the heparin sulfate is acting as an initial attachment factor, and that it's not directly decorating the ACE2, and that it binds to a distinct part of the spike protein, and that that initial attachment is sort of like potentially handing off the virus to ACE2. So in that sense, it may be cooperative. I don't think that ACE2 is directly sulfated. There have been some nice binding studies. The structure that I showed there, I neglected to reference where that structure came from, but I can send it to you after. There are a couple of nice papers from, I forget the name of the group, but it's at the Scripps.
Okay, thank you.
Yeah.
And we have a question from Rodney Rothstein. Rodney, you should be able to talk.
Yes, very, very nice talk. I wanted to ask you a question about positive and negative interactions, because protein-protein interactions don't tell you whether something is going to be positive or negative. Do you have any, most of the things that you talked about were negative, because if you knocked out the host function, it disrupted the virus. Did you find any positive ones?
Almost all the that I can recall from the protein-protein interaction network, all of them looked like they gave it a selective advantage when they were knocked out. We did find some interesting negative regulators from the genome-wide screen. One of them is a phospholipase scramblase that we don't quite know what it's doing, and we've found that this is kind of, this rhymes with a lot of the work that we've been doing of studying proteins where it's not quite clear what the function might be. There are also STAR-D3 and STAR-D3-NL, which also have a role in lipid transport. It's interesting that the role that they play seems to be in the opposite direction of some of the other regulators of lipid metabolism that we've seen. So certainly the functional host factors that we identified went in the direction of being pro-viral.
Okay, we have a question from Steve Goff. So Steve, please go ahead and unmute yourself.
Yeah, hi JT. So, we think a lot about the readouts and the selections in those screens. So you're looking basically at an enrichment, of guide RNAs in the pools. So you're in a sense looking at, I guess, cell survival, with virus killing. And I wondered if and we do those too. And the alternatives are to look more directly for virus load or virus replication rather than cell death versus cell survival. And I guess that's your secondary screen was more for virus replication sometimes as opposed to cell death. So I just wanted to see what you thought was best, or what's the most effective screen?
Yeah, I think they're complementary. Definitely the easiest screens to do are the survival screens. And, we found that that was the screen that was tractable at sort of the early phases of the pandemic when there really, there wasn't for example, another type of screen that I think you're alluding to is you could use a reporter virus and that reports on infection instead of survival.
Exactly.
And, when we started these screens, that didn't exist for SARS-CoV-2. So it wasn't a tractable screen, but I think it would save a lot of work to do some of the things that we have to do at the endpoints of these screens, which are, bypass binding by transfecting in infectious RNAs with reporters and things like that. So I think that they're complementary. One of the screens that's published now that I whizzed by basically did a great screen looking at trying to find a CRISPR-I screen to identify host factors that support ACE2 expression using a FACS screen with a purified receptor binding domain. I thought that was really beautiful. And I think that these kind of multi-level screens can really help you deconvolve like survival-based factors versus factors that are really important for the life cycle of the virus. One other screen that I went by here very quickly from the Yale Broad collaboration did some of the screens using pseudotyped viruses, which was pretty interesting. And that really also gets right at the entry portion of the screen. So, there are a lot of ways to go after this. I don't think any one specific way is best, but some are more tractable than others at specific times.
Right. And I think there can be interesting discord between them sometimes where, you might get, protection from cell death without, impacting all aspects of the virus. And those are interesting too. Yeah.
And I'll say that we did our primary screens in one cell line, and other people choose different cell lines from different species. And it's been interesting to compare the results because sometimes you find things that are in common, other times you find things that are very different. And so now we need to kind of start to deconvolve what the basis of those differences is.
Right. No, I think there's going to be a lot of that where there are, especially where the virus has alternate entry routes, for example. So in some cell line you may block and another cell line not.
Yeah, that's true. And it's one of the things that we're starting to dig into a little bit with ACE2. Can you make a pure knockout population insert in a cell line that is naturally susceptible to ACE2, or excuse me, to SARS-CoV-2, and will that in any way support infection of the virus? And I think there'll be some interesting emerging data on that.